Hello, in this video we will discuss about the ethylene absence and presence signaling pathway and what will be changed. So this is the plant which that is used for the photosynthesis to development and growth. So this is the plant photosynthesis, and the stem is used for the transportation while the leaf is used for the photosynthesis. So the primary role of Yang cycle in the leaves of higher plant is the for the synthesis of its ethylene, recycling of MTA and produced during the polyamine biosynthesis in the leaf vasculature. So the ethylene signaling is a signal transduction in plant cells to regulate the important growth and development processes, acting as a plant hormone. So it is the gaseous stage, colorless, tasteless, and it will produce and will trigger the germination of seed. It means the germinate of seed, ripening of fruit. It means the development will be decreased while the ripening, and growth will be decreased while the ripening will trigger. So the smaller fruit can also ripe. So opening of flowers and abscission and shading and stress response. This is all thing is due to the ethylene presence. While first of all we will learn about the cell structure, which contain the outermost layer is the cell wall, inside is the plasma membrane, while the Golgi bodies and the endoplasmic reticulum is present. So remember that things. And this is the cytoplasm, and this is the endoplasmic reticulum. This is the nucleus, and the endoplasmic reticulum we will understand because it contains a subfamily 1 and 2. This subfamily 1 and 2 is outside is the lumen, basically I am showing in the illustration, while the inside is the cytosol, so the cytoplasm. So this contain ETR1 and ERS1, while the subfamily 2 containing are ETR2 and EIN4 and ERS2. This is basically the name of this is the ethylene response receptor 1 ETR1, ethylene response sensor ERS, and ethylene insensitivity EIN4. So the isoform of ethylene receptor in Arabidopsis thaliana, which that is we are discussing in this video about that exemplification of this plant, and the different plant contain the different type of receptor and responses. So let's begin to understand the absence and presence of ethylene, the signaling pathway, the changes and how it is produced and what will be changed, because the no ethylene will lead to develop and growth while no ripening, while the presence of ethylene will lead to ripening of fruit and seed germination, etc., etc. So this is the nucleus and the endoplasmic reticulum layer. I am drawing just for illustration. The outside is the lumen while the inside is the endoplasmic reticulum, and inside is the nucleus are present, which that is same. The illustration is difficult to understand, but you can understand. So the no ethylene response first we will discuss. So endoplasmic reticulum is this, and no ethylene response basically the RAN1, which that is the calcium efflux from this channel, will lead to phosphorylation in the ETR1, ERS1, ETR2 and EIN4 and ERS2, which that's collectively will trigger the EIN2, and EIN2 will in the presence of CTR1, the constitutive triple bond response 1, which that will trigger the EIN2. EIN2 triggering in the presence of ETP1 and 2 and SCFE2, which that will trigger the EIN2 phosphorylation, will lead to ubiquitination to proteasome formation. This proteasome is basically used for the growth. Proteasome, so proteasome means the protein synthesis for the development and growth, but not ripening. So remember, inside, in the absence of the ethylene, no ethylene stimulation will lead to produce the messenger RNA, the 2 mRNA and EBF1, which that will be transcribed to translate First of all, we will need to understand this is the EIN2 and this is the phosphorylation. EIN2 phosphorylation and ubiquitination lead to proteasome activation, and this proteasome activation also triggered by the no ethylene stimulation in the DNA. So the DNA will transcribe. Let's begin to understand also the DNA transcription. So this is the nucleus contain DNA in the form of chromosome. So the active part of the DNA which that transcribe that gene in the absence of ethylene, the no ethylene response. So the no ethylene stimulation will lead to EBF1 and 2 mRNA, which that will be triggered and move into the cytoplasm. So this is the no ethylene stimulation. After the no ethylene stimulation, the EBF1 and 2 mRNA, which that will trigger and go into the cytoplasm, the SCFE3 and FBF1 and 2. This FBF1 and 2 and SCFE3, which that's collectively produced, and in this way the EIN3, ETL1 and EIL2. This is the protein which that will be stimulated via a FBF1 and 2 and the SCFE3 to ubiquitination, the EIN3, ETL1 and EIL2, which that will lead to proteasome production, and in this way the ripening will be inhibited. So remember that things. While in the case of ethylene, so the ethylene presence will inhibit the ETR1, ERS1, ETR2, EIN4, ERS2, which that will lead to CTR1 constitutive triple bond response 1 phosphotransferase enzyme. In this way, the ETR1 phosphorylation, the cascade of phosphorylation will lead to active the AHP, the phosphorylation of the AHP, and this AHP will be phosphorylase go into the nucleus in the DNA, binding with the DNA. Remember, this is the AHP phosphorylation. After the phosphorylation, the AHP will go into the nucleus. Remember. So on the other hand, the EIN2, is the ethylene insensitivity, which that will lead to protease enzyme will lead to EIN2C formation, and in this the EIN2N, which that will be also present. So the mRNA, the EBF1 and 2 will produce when during the no ethylene stimulation, so that when ethylene are present, so in this way the FBF1 and 2 and mRNA, which that is basically in the presence of the ethylene, the EIN2C will bind within it the messenger RNA, while it will not bind if the absence of that ethylene. So EIN2C complex formation with the EBF1 and 2 and mRNA P body, which that will lead to transcribe and will lead to use for that synthesis of protein for ripening of fruit and storage of glucose. So this is ethylene stimulated transcriptional regulation through AHP will be phosphorylase, the ARR. ARR phosphorylation will lead to also produce that proteins and storage a mechanism of that glucose into the fruit will lead to ripening of fruit. The color will be changed, and that is why the ethylene presence. While in this case here is the absence of ethylene, RAN1 will calcium efflux will trigger the ETR1, ERS1, ETR2, EIN4, ERS2. While on the other hand, the inhibit this in the presence of ethylene. So remember that thing. So ethylene response will lead to fruit ripening and fruit maturation, and that is why growth will be stopped and the development will be stopped while the ripening will trigger. By this stimulation, transcriptional regulation to produce and synthesis that proteins in the fruit will lead to ripening of fruit. So that is the process in which the uh, signaling pathway of the ethylene presence will lead to ripening, while the absence will lead to trigger that ERS2, EIN4, ETR2, ERS1 and ETR1 will trigger the EIN2, lead to growth but not ripening. So thanks for watching. Please make sure to subscribe. Bye.